Assalamualaikum warahmatullahi wabarakatuh. I hope everyone have a nice day today. This is the video presentation about my final year project entitled Biodegradation of Microplastic by Microorganisms in Marine Habitat, a Review. Before I start, I would like to introduce myself. My name is Nurshahirah binti Sazali. My matric number is 2016970141. For your information, I was supervised by Madam Atika Husna binti Ahmad Nasir, and my co-supervisor is Miss Wahana binti Osman. Before I start my presentation consists of introductions, issues and problem statement, significance of study, objective of study, research questions, literature review, future research, conclusions, and the last one is references. Now I will move to the introduction part, which is microplastic that have been broken down into smaller pieces can be transferred into the seas from waste dump and contribute to microplastic contamination. This part will be supported by Aloma Etta that said that account for 60% of the estimated 8 million tons of plastic entering the world's ocean each year. So, marine bacteria and archaea also greatly contribute to the respiration and cycling of several components of the surface ocean. So now let's move to the issues and problem statement. The problem statement that I have been stated here is the Strait Times in Singapore state that in Malaysia, the most number of the plastic was dumped, a small amount was burned, and only a little was recycled. And in marine habitat, the problems lie when the most of the marine life have mistaken that the plastic is their food and accidentally eat the plastic that causes suffocation and starvation and drowning. So we can see here that there are many plastic dumped into the oceans, and for the marine habitat and marine life, they cause suffocation, starvation, and drowning. So this make an issues for health issues and environmental issues. For the health issues, toxic chemicals from plastic could impact human health, such as hormonal birth and cancer. For environmental issue, the high surface area to volume of micro and nanoplastic has accumulated with the other pollutant, thus cause virulence into the aquatic environments. Next is the significance of study. As we can see above, there are microplastics can be found worldwide in aquatic ecosystem reaching from marine to freshwater region and from populated to pristine. And the definitions of microplastic remain vague, and different size classes are investigated in monitoring the studies. Next, I will move to objectives of study. For the first objective is to identify the biodegradation capability of marine microorganisms to micro and nanoplastic in marine organisms. So basically here, when there are many marine life that suffer because of the plastic, actually from the macroplastic then turn to the microplastic and also will turn to the nanoplastic. Basically each of the macro, micro and nano give an impact to the marine organism. Then how we want to reduce this by find or identify the capability of marine microorganism in this paper. And for the second one is to suggest possible treatments and method on overcoming plastic waste in marine habitat. So basically here, the objective is one to find the possible treatments or method that we can use it to overcome the plastic. So how I want to stick with the objective, I've made some research questions. For the first one is, what extent does exposure to micro and nanoplastic lead uptake in marine habitat? Second is, why microorganisms used to degrade the plastic waste in marine habitat? And the third one is, what is the best treatment and method to overcome plastic waste in marine habitat? Actually, for the there are a few microorganisms that are used to degrade the plastic in my habitat, but in the treatment or in the method, there are still many research to do. There are a few treatment and method, but it quite pricey and the cost is too high. So basically, I've done with my introduction part, objective, significance of study, research questions, and now let me move to the important part, which is literature review. So basically, what we can see here, the differences between microplastic and nanoplastic. So this table show the differences between both microplastic, known as primary MPS, and mostly observed in exfoliant like hand or mouth cleaner. And for example is toothpaste. For nanoplastic, we can known as primary NPS, mostly observed in laundry of clothes and the disintegrations of artificial fiber. And the examples is detergent. Like I just said before, there are a few types of microorganisms that we can use in the biodegradations. For the first one is Bacillus subtilis, and the second one is Ideonella sakaiensis. For the Bacillus subtilis is the one that can survive in different conditions. So there is no problem for them to stay in everywhere, whether in land or oceans. For the second one is Ideonella sakaiensis 201 F6 degrade 18 PET. So Ideonella sakaiensis is one of the microorganisms that can be used to degrade the plastic. So as we can see here, there are a few steps involved in biodegradations of plastic in marine. So for the first step is microbial biofilm formation. Actually, fungal biofilm is an association of microorganisms in which microbial cells adhere to each other on living or non-living surface within an EPS. EPS also known as extracellular polymeric substances. And for the biodegradation is any unwanted alteration in the properties of substance caused by the essential activities of the organism. For the third one is biofragmentation. The biofragmentation is actually the polymeric material will broken down and turn into the oligomers, dimers, and also the monomers. And for the last one is the mineralization. Mineralization is the degradations of synthetic polymer pathway in the aquatic environment with the participations of degradation process. It will can be the intermediate step um, before they want to complete the mineralizations. So basically in this picture there are illustrations of plastic biodegradations by microorganisms. So far as we can see in the first picture, there are initial attachment of microbes on the plastic surface. The polymeric material that can be turned to the microbial biofilm formation, and the next step is biodegeneration, which is secretion of extracellular enzyme and EPS, and biofragmentation formation of oligomers, dimers, and monomers, like I just said before. And for the last one is mineralizations or microbiomass that consists of carbon dioxide and H2O, that water and carbon dioxide. And we proceed to the last but not least, my future research. First is protein engineering of enzymes. Second is strain engineering. Third is in silico genome mining. Fourth is metagenomic analysis. For the protein engineering of enzyme, actually used to remove the microplastic accumulations. For the second one is strain engineering, the technique of genetic engineering that used to create a very successful microbial strain to degrade synthetic polymer. And the third one is metabolomic data will move the predicted molecule more effectively and vice versa. And for the metagenomic analysis, this is the shotgun metagenomic used to sequence of plastic microscopes following biofilm and suggested enrichment of separate enzyme. Basically here, there are a few future research that we can use to degrade the plastic. But this, like I just said before, this waste and research, we can use it, but the price is too pricey and quite high. So for the last one is the conclusion part, which is the conclusion part is to answer my objective part. Basically, this analysis conclude that the exposure of microorganisms like Bacillus subtilis and Ideonella sakaiensis have particular enzyme. Basically, as we can see here, both of them, like Bacillus subtilis and Ideonella sakaiensis, is the microorganism that have particular enzyme that used to degrade their polymer, which is they can use to break the structure of its back of their backbone and also its side chain. So basically, we can see that synthetic plastic waste is very dangerous to us and extremely can give the biodegradation resistance and also causes a numerous environmental problem for like suffocation, accumulation, and long-term toxic effect. We can see that we didn't want to use the synthetic plastic waste, but in the short term you have to reduce it time by time so that we can save the marine species in our oceans. So basically in this slide there are a few references about my review paper. So thank you to my supervisor, my co-supervisor, and also my panel to see my video. Stay safe, stay at home. Thank you. Assalamualaikum warahmatullahi wabarakatuh.